The overall goal of this procedure is to create a model of acute kidney injury in zebrafish using the aminoglycoside gentamicin as the nephrotoxin. This is accomplished by first injecting gentamicin into the common cardinal vein of 2 days post-fertilization zebrafish larvae. The second step of the procedure is to score for edematous phenotypes in 4 days post-fertilization injected larvae. The third step of the procedure is to process 4 days post-fertilization zebrafish for immunohistochemistry, staining with an antibody for kidney tubule epithelium. The final step of the procedure is to section zebrafish larvae and visualize for damaged tubules due to loss of cell polarity and disruption of tubule structure. Ultimately, results can be obtained that show damaged renal tubule segments through immunohistochemistry with a kidney epithelium antibody.
Hi, I'm Chiara Cianciolo Cosentino from Neil Hukriede laboratory in the Department of Developmental Biology at the University of Pittsburgh. Today we will show you a procedure to microinject the aminoglycoside gentamicin into 2 days post-fertilization zebrafish larvae. We use this procedure in our laboratory to induce acute kidney injury in larval zebrafish. So let's get started.
Start by breeding adult zebrafish 3 nights prior to the planned experiment to stage embryos at 2 days post-fertilization. Prepare E3 medium using 5 millimolar sodium chloride, 0.17 millimolar potassium chloride, 0.33 millimolar calcium chloride, 0.33 millimolar magnesium sulfate. Collect embryos and place them in a petri